Hello students, today we are going to discuss about the what are the role FDA play in drug discovery process, right? So when we talk about the NDA, new drug application, as the name indicate, application means applying of the approval of new drug to the FDA, Food Drug and Administration, that is one of the governing body which will be having all the country, all the reason for the approval of the drug. So FDA is going to check the obtained evidences in the form of data, whether it is significant, safe and effective or not. Then they are going to give the approval. So let us discuss one by one. In first pre-discovery, first when we talk about the pre-discovery, the pre means before discovery, the what is going to happen whenever we talk about the drug discovery, right? So drug has to be there into the particular natural origin. It has, right, it will be there in the, it may be in the plant kingdom, it may be in the animal kingdom, it may be in the microorganisms, minerals, whatever may be the natural source where we are thinking that there may be the particular types of chemical entity present which may show the activity onto the particular disease and disorder, right? Then accordingly that we are going to select, and the study will go on like extraction, isolation, characterization, different chromatographic techniques, spectroscopic techniques. Then we are concluding with the particular structure, that this particular structure, it is having analgesic activity, anti-inflammatory, anti-diabetic, hepatoprotective, or what kind of activity, right? So once the discovery will complete and we will conclude this particular chemical entity is having effect on that particular types of issue or elements, right? Then it will go to the preclinical study, right? Second step, in preclinical study we are going to select the animal model, the animal model which is anatomically and physiologically resemble to the human volunteers or human body, right? So that we can expect that whatever the result we are going to get in preclinical or into the animal body, the same kind of result we can expect into the clinical trial or in the human body. So in preclinical trials we are selecting the particular rats, mice, guinea pig, monkey, microorganisms, parasites, earthworm, it may be anything, depend on to the study. Say for example, if you are going to check the antimicrobial activity of the particular drug or particular chemical entity, we have to select the particular strain or species of the microorganisms like bacteria. It is for antibacterial means bacteria we have to select. If it is antifungal means fungus we have to select. Anti insecticidal means insects we have to select. So if it is anti-diabetics means Wister albino rats we have to select. So for example, in antimicrobial you have selected the particular bacteria like E. coli. So we are going to check our tested drug or extracted isolated drug onto the bacteria, whether this particular tested drug or extract it is able to kill the bacteria available on the particular sample or not, and what is the level of significance the giving the killing activity, right? If it is anti-diabetic, we are going to select the Wister albino rats. In that rats we are inducing the diabetes, right? The sugar level into the blood and urine. So we are giving the streptozotocin or alloxan induced diabetes. There are so many animal models are available. So once the diabetes will increase, we are giving our tested drug by oral route or different other routes, and we are checking the blood, we are collecting the blood sample and checking whether the particular tested drug it is able to reduce the sugar level onto the blood and urine or not. If it is for hepatoprotective activity or liver protection, so first we have to damage the liver of the particular rats, right, by giving the allyl alcohol or paracetamol. Different models are available, and once the liver will damage, we are giving our test drug in the preclinical, so that we are going to check that whether the particular drug is able to heal the hepatocytes or increase the number of hepatocytes or not. That's what we need to sacrifice the rats, and we are collecting the blood samples also and time to time, and we are checking SGOT, SGPT, total bilirubin, cholesterol, and all those parameters available. And at the same time we are going for hepatocytes, the microorganisms, the microscopical examination. We are checking the degree of damage or healing, rate of healing, and all those things, right? So this happen in preclinical. Once you are getting the significant result in preclinical, the drug will go to the clinical trials. The clinical trial, it is divided into the phase 3 study, phase 1, phase 2, and phase 3, right? Here, remember, in clinical trial we are taking the human volunteers, right? So here the number of volunteers, it is going to change in each phase. In say for example, here, that's what in clinical trial, IND submitted, right? What is IND? Investigational new drug, right? Whatever the drug it is under investigation, that drug will go to the phase 1, right? So once in phase 1, 20 to 100 human volunteer, healthy human volunteer we are selecting, and we are giving those volunteers to that particular drug at the suitable route which has been identified, and we are checking whether the drug is giving any kinds of side effect or not, any kinds of adverse drug reaction or not, contraindications, drug interaction, drug food interaction, what kind of activity it is having in overall. Coming to the phase 2, once you are getting the satisfactory result into the phase 1, the drug will go to the phase 2. Here the number of volunteers is going to increase 100 to 500, right? Minimum is 100 and maximum is 500. Here, once again, the drug will be given to these human volunteers, and we are going to check the again the effects of the drug. And once you are getting the satisfactory result, it will go to the phase 3. Under phase 3, the number of volunteers, it is going to increase 1000 to 5000, right? In this high range of the volunteers, we are giving the particular drug for the particular disease, right? Whether if it is anti-diabetic, so diabetic patient, we are going to select these volunteers should have the diabetes, and they are going to receive the particular drug. And we are going to check the whether the drug is able to reduce the blood sugar level or urine sugar level or not, right? What for it is. So this process, it takes 6 to 7 years of the time, right? Here in the drug discovery and preclinical may take 3 to 6 years, right? Coming to the NDA submitted, right? New drug application submitted here. Once the phase 3, it will pass, the drug will pass phase third phase also, then it will go for the review, right? So FDA, the Food Drug and Administration, is going to review the obtained evidence, obtained data from the right from the beginning, from drug discovery, preclinical and clinical trials. And once they will get the satisfactory result, they are going to give the approval. So FDA approved the drug. So then the scale up to manufacturing, the manufacturing will start here, right? It will be incorporated into the suitable dosage form so that it can be launched into the market. This process will take half year to 2 years of the time. Then after this step, it is going to have the post marketing surveillance or phase 4 study, this is called. Once the drug will launch into the market, then doctor will start the prescribing in the phase 4. Then patient will start going to take this one, and after that there may be the chances of giving having the particular types of side effects or adverse drug reactions, right? So that's what here the time is indefinite, right? After drugs, it is existing into the market, it may be 10 year, 20 year, 50 years also. There may be, so it may show the particular types of adverse drug reactions. So after that also drug may be banned. The FDA is going to ban that particular drug. That's what so many number of list of the drug has been banned by the US government and India government and different other countries also, right? So that's what the time it has not mentioned, indefinite time it has given, right? So like that it will go on. So yeah, that is the role of the FDA into the new drug. That's what now the vaccine, it is going on in India for COVID-19. So it has if you will talk about this time duration, 3 to 6 years. So take the highest example, 6 years, then here 6 to 7 years, then here zero point 0.5 to 2 years. So if you will calculate this 6, 7, 13, 13 and 2, 15, 15 years minimum it will take to come into the market of that particular drug. So once it will come into the market, the 15 year, what will happen? It may That's what in vaccine it has come almost in 15 month, right? So in emergency case, they are going to skip or predicting that particular phase, and they are launching in the drug into the market, right? So that's what if you go proper way, and if the situation is not emergency, it may take this much of time, right? So that is the role of FDA in the drug discovery of new medicines.